enough so that they can activate transcription of the reporter gene so that you would see, in this case, growth on histidine. If those two proteins don't interact, then you're not going to have any transcription. So that then there's no transcription here, there's no gene product, there's no protein, there's no growth on histidine. Right? So if you were to make this type of construct and you had two proteins which you thought might interact, then this would tell you if they do or not. Right?
The previous example, you just insert the existing gene or just take a promoter?
Right. So in this case, this is yeast, and this would be a whole construct that you would put inside the yeast. So inside the yeast genome, you would put this whole construct. You would put the right, essentially just this piece. You'd put the upstream activation sequence. You'd have the promoter, and you'd have the reporter gene. And then you would have genes encoded for the proteins of interest. So you'd have a gene that encoded for your one protein fused to the binding domain, and another protein fused to the activation domain.
How do we know that they don't interact? They're trying to remove just couple with the activation domain without double quality.
Well, you don't only through experimentation. So in the test tube, you could get it to work because you can make the concentration high enough. So it's true that the A domain here, in this case, the DNA binding domain and the GAL4 activation domain, if they're independent of each other, they'll drive transcription. But at this point, when two proteins, two regions, two domains of proteins are on top of each other, their concentration and relevance to each other is essentially infinite, right? So as soon as you take them apart, they have this concentration dependence because this protein and this protein have to diffuse in order to get into the same region with each other. So in vitro, in a test tube, you could conceivably drive transcription because you could be able to control the concentration of the two domains. But in a cell, it's going to be a rare occurrence that a non-fused proteins go into the proteins once they're apart, that they're going to find each other. They're going to find each other much more rapidly if you have two proteins that obviously interact with each other. Oh, yeah. And then the third way, looking for protein-protein interaction, is called phage display. So bacteriophage are viruses that infect bacteria. So one of the uses of this, or one of the good things about being in bacteria, is you can make lots and lots of them, right? So it's really easy to make phage and produce many, many phage particles. Okay? What happens here is you have phage. Phage is a virus, so viruses very often have a capsid, protein capsid. The protein capsid has proteins which are exposed to the outside of the cell, right? Inside a viral capsid, in this case, there's be a picture, but in this case you have a virus that looks like this, right? Inside the DNA, and then all around the outside is the viral capsid, the protein envelope that encases the DNA. And in this case, there are some specific proteins that are on the outside of the virus. And similarly to the yeast 2 hybrid assay, you can make fusions of whatever protein or peptide or oligo that you want on the end of one of these proteins which are on the outside of the virus, exposing it to any potential interactions. So there's a picture of what I was trying to draw. So this is the virus, the viron, this is the outside protein envelope, capsid, and then you have proteins on the outside that are sort of exposed to the environment, and you can fuse them with different types of peptides or proteins, okay? And making very many different kinds of combinations, okay? So that you can then put these with another protein and see if there is any binding. So in this case, similar to tag, you would put maybe a protein of interest and you put that in an affinity column. So you'd have beads with your protein bound to those beads, right? And you want to know what peptide binds to this protein. And so you say, I want to know what combination of, say, 12 amino acids binds to my specific peptide, right? So you make, inside bacteriophage, you put your protein envelope or capsid protein, and then on the end of that, you put every combination of 12 amino acids there is. Okay? You make that many viruses. You make every virus that represents every combination of 12 amino acids on the end of this protein, envelope protein. Okay? And then you pass those bacteriophage over the column. And any of the bacteriophage that bind to that protein will stick to the column. And then once you wash those off, you can infect bacteria and grow them up. And now you have huge amounts of the peptide that you know bind to your protein, and then you can do protein sequencing, see their, say, end-terminal degradation, or you can do all the top, something like that, to get the identity of the amino acids that now just bound to your protein of interest. Something else that they do is they make, so when I said that they say you would make phage